Okay, we're going to go ahead and get started. We have a very special session of office hours today to do an overview of our newly released all by all data set. And we're excited. It's been a long time coming, and we're excited for you all to learn more. Before we get started, I do have a few announcements before I introduce our speaker today. So in case you missed it, we had our last office hours about SAS Studio that is now posted on our YouTube channel and the User Support Hub. So if you wanted to go back and review all the information that was provided by our SAS Studio team, feel free to scan the QR code to learn more. And I'll give everyone a moment to do that. And then this is just a quick announcement about the release of our all by all data. So all by all tables are available in the researcher workbench for controlled tier access only. So if you are a registered researcher of the researcher workbench, you must have controlled tier access to access this data set. That being said, we do have a user support hub article that gives a broad overview of the data set, the data format, and the QC steps that Wenhan is going to go through today. So please visit allofus.org/allbyall or scan the QR code to learn more. So I'm going to introduce our speaker today before I pass it off to Wenhan. So Wenhan is a senior computational associate for Dr. Benjamin Neal and Dr. Conrad, I always butcher the name, Karskowski's lab at the Broad Institute. Her research focuses on developing methods and pipelines to process large-scale sequencing call sets and statistical methods for interpreting the impact of rare genetic variants. She earned her master's in biostatistics at Yale University and really was helping pilot this launch for the all-bio dataset. So we're excited to hear from Wenhan, and I'm going to stop sharing my screen, Wenhan, and pass it on over to you.
Sounds good. Thanks for the introduction. Okay, so hopefully everyone's able to see my screen right now. Okay, I'll just get started. So yeah, I'm today very excited to introduce our all by all project, which encompasses the common and rare variant association tests in about 250,000 whole genomes across diverse ancestry groups from the all of us data. And then the goal of my today's presentation would be a very quick walkthrough of how the data did come into being. And also we can together take some close look at the snapshot of those results. So since the data since this all by all project is completely based on the data from all of us, I think I would like to briefly recap some of the features of the all of us data before we dive into the results. So a lot has been achieved by the All of Us research program through the past few years. By 2023, there has been over 750,000 consented participants joining the study, and then we have 250,000 individuals with their whole genome sequencing data being generated. And then the data has been widely used by researchers all over the world. And not just the genotype data, the All of Us research program has also collected a wide variety of phenotype data, including about over 400,000 surveys responses, 300,000 physical measurements, and also those comprehensive EHR data, which can be later curated into disease codes that are super helpful in association testing and then gene discoveries. So in addition to the wide range of genotype and phenotype data, the All of Us cohort is highly diverse in their genetic ancestry groups. So among the 250,000 samples, they're from 6 different ancestry groups, where we have a large proportion of AFR and AMR ancestry groups. Those people are super valuable in increasing our diversity in genetic research. And then the whole genome sequences covering hundreds of millions of variants will enable us to implement the GWAS and the rare variant association studies across the many available phenotypes simultaneously. And with that being said, we were able to build our GWAS and RWAS pipeline and then set our all-by-all analysis from here. So before running the association tests, we did some initial processing of the phenotypes and the samples to both obtain some additional information that can be used for later association testing and also ensure a high quality of the data. So on the sample end, we had several sources of information, which are demographic information, relatedness information, and also the genetic ancestry groups. So just as a quick recap, the number of samples from each ancestry groups are original as such. And then from there, we were able to annotate the covariates that will be used in association testing, such as age squared, sex, and then some interaction terms. And then using those information, we decided to keep samples with sex defined as female and male, age below 100, not any within the duplicated sample groups, and also with their ancestry category defined. And then on the phenotype side, we have 6 phenotypic categories in total. So for those binary ones, we have about 1,000 prescriptions, about 100 personal and family history surveys, and also like about 5,000 phi codes in total, which include both phi code and phi code X. And for the quantitative trace, we have 7 physical measurements originally, and then from there we derived 3 customized phenotypes, which are BMI, weight heap ratio, and weight heap ratio adjusted by BMI. And then we also had 6 lab measurements. So in total, we have 70 quantitative traits. And then using those information, we were able to define sex-specific phenotypes for those phenotypes with more than 90% of samples from one sex category. And we also computed the case control information and then annotated the actual definition of those phi code in the prescriptions. So then we combine the sample information and the phenotype information all together within in a combined set, which can be later used to generate the small phenotype text file being used for association testing. And then to ensure we are running powered enough tests, we also filtered phenotypes to those with number of cases over 200 per ancestry groups. So from this table, you can see that among those 6,500 phenotypes I just showed in the previous slides, we ended up with about 3,400 unique phenotypes that passed this criteria. And then in this table, we also break down into each phenotypic category and then present the number of phenotypes across each ancestry groups. And then here, we also checked for each phenotype how many genetic ancestry groups it is defined across. So as you can see, there are about 100 phenotypes that are well defined across all 6 ancestry groups. And then we have about over 3,000 phenotypes with more than 1 ancestry groups defined, which gives us enough information to implement meta-analysis after we run all the association tests so that we have enough ancestry groups to combine together to implement a better powered meta-analysis. And in addition to that, we also took a look at those phenotypes that are shared between UK Biobank and all of us. So here we're comparing the prevalence of those binary traits shared between the 2 cohorts, and then we can see that those phenotypes, the prevalence of those phenotypes are pretty much concordant within the 2 groups, and especially for those 3 ancestry groups with more phenotypes shared. especially in the EUR category, we can see that we are having phenotypes with higher prevalence in the all of us groups, which is pretty nice because we can have like a better power here by testing those phenotypes. Yeah, so here we're pretty much showing the good concordance between of the prevalence between UK Biobank and all of us. And then after curating the phenotypes, we also did an additional filter on the samples to reduce stratifications, because within one ancestry group, if there's like a super admixed structure of samples within the single group, it will cause problems when we're later computing our genetic relation matrix and then implementing our association test. Most often it will introduce some non-converging issue. So here we're using this pipeline to reduce stratification by first starting from the ACAF matrix table, which is the common variant matrix table from all of us. We split it into the related and unrelated samples, and then filtered to a million high quality variants, and then run our own population or ancestry specific PCAs here. So we run PCA within each ancestry group, and then we project those related samples onto the PC loadings of those unrelated loadings. So here we get a new set of ancestry specific PC scores, and use that. We implement a pipeline called centroid pruning, meaning that we remove those ancestry outliers based on their multidimensional centroid distance computed from their average PC values. So therefore, we have a set of samples with those outliers removed, ended up with a more homogeneous set of samples, and then there we there's something missing up here, but yeah, we then compute another round of PCA on those pruned samples, and then ended up with the final PC score to be used in association testing. And then from this table, you can see from the original sample size, we pretty much preserved 90% of the samples within each ancestry groups. And then here, I'm gonna introduce to use like a quick small pipeline where we generated some random phenotypes. So as some of you might have already noticed in the previous phenotype table, there are like 30 random phenotypes included, which is basically generated by this pipeline. So here we aim to produce a set of basically null phenotypes, which shouldn't have any genetic associations, to be used as a tool to tell how well our test is performing. So to start with, we use the exome matrix table from all of us, and then select a representative subset of the variants for each ancestry groups, and then subset the matrix table for each of these ancestries. And then from there, we run LD pruning and then generate the plink files for each of them. And then we use SAGE, which is the association testing software we're using later. And also it has a step 0, which can be used to generate the genetic relationship matrix. So for each ancestry group, we generate this GRM and then load this GRM into R using this function called RMBN sparse. It takes account the genetic information from the GRM and then generate a set of random simulated phenotype from the multivariate normal distribution. And using those simulated phenotypes, we're able to pre-specify some prevalence, heritability, and then the features of the phenotypes we want, and then just use some quick linear combination to construct the random phenotypes we wanted. And then those phenotypes are also included in the results. And we also had like association test results for these. Okay, so with all the infrastructure being described, here is our gonna be our full pipeline. So to start with, we use the genotype data from all of us, which include the exome matrix table and the ACAF matrix table. So the exome matrix table is basically include all the exonic variants. And then the ACAF matrix table is filtered by the ancestry-specific allele frequency and allele counts. So it includes those so-called common variants. And then from there, we prepare our input files to the association test pipeline. For the genotype information, we just truncate those into small intervals so that it's going to be easier to run in parallel. For the exome variants, we trunk each interval to be those variants within about 200 genes. So we ended up with 99 bgen files per ancestry groups. And then for the ACAF matrix tables, we did this truncation by specifying the length of the intervals. So we did a relatively smaller interval for the EUR and then for a relatively larger interval for non-EUR. So we have 73 intervals for non-European ancestries and then about 136 intervals. And some other input files, including the phenotype file I just described, we just select for each individual phenotype and the ancestry group, we just export all the information related in a text file. And then from the combined data table I just described. And then we also use the Plink file in the GRM described in the previous slides when we're generating the random phenotypes. And then here we run our association test in Sage. It consists of 2 steps. In the first step, we used the phenotype and the subsetted genotype information from the Plink file in the GRMs. And for those exome, for the exonic variants, we specify some arguments to make sure that we include the variance ratios for both common and rare variants. But for the ACAF part, we only specify some several common variant categories to increase efficiency. And then once we've completed step 1, we will have a null model for each phenotype. And then in step 2, we load the null model and the truncated bgen files so that it has enough information to implement the association tests. So in the end, we will have 3 different types of association tests. The first one is the gene level results generated from the burden test, which is basically grouping all the variants within a gene of the same annotation category together as a whole, and then test them against each of the phenotypes available. So in this table, you will see the association test is between gene and a gene annotation group versus a phenotype. And then for the other 2, those are the single variant results for the exonic variants and also the common variants from the ACAF matrix tables. And each of those results are available for all 6 ancestry groups with those population that has enough number of cases. And then from there, we meta-analyze the result from all 6 ancestry groups and then end up with the meta-analysis results for the 3 types of association tests. And with that, we first examine our association test result with our random phenotypes. So we evaluate the overall performance of GWAS using this term called lambda GC, which is the genomic inflation factor. It indicates how well calibrated the association tests are. So if it's closer to 1, it means that our association tests are behaving pretty well. And as you can see from the QQ plot, each dot represents 1 genetic component, and then so it represents the p-value of this genetic component versus the phenotype on top of this panel. So as you can see, for those random phenotypes with a prevalence over 20% or like very high prevalence or continuous trace, the test summary stats are pretty well calibrated, especially when you can see the dots are all on this dotted line y equals to x, and all the lambda GCs are super close to 1. But then if you take a look at those rarer outcomes, especially when the prevalence dropped to below 1%, we start to see some underpowered, deflated results, especially if you can see here for the mid and the SAS ancestry groups, those results are looking super off. But since we have previously done this cutoff of phenotype with number of cases over 200, and then given this, we're pretty confident about that cutoff and the general performance of our association tests. So now we're good to go running the association tests on all the real phenotypes from all of us. We here have some summary stats on the technical end. So in total, we have about 8,900 phenotype ancestry group pairs, meaning that we run 8,900 GWAS and RWAS, and then 7,800 of those are from the three largest genetic ancestry groups, meaning EUR, AFR, and AMR. With all those BGen chunks, the small genotype information files I just described, times all the phenotypes we're going to run, we ended up running about 2.7 million jobs, and then they took about 845,000 CPU hours. So this humongous pipeline was implemented in Hail Batch, which is like a Python, which is a model within the entire Hail Python module. It makes the pipeline super efficient by making it scalable on the cloud. And then in this case, we can run tens of thousands of CPUs all at once. So we can run this 2.7 million jobs in parallel, and then you don't have to like wait for a long time before you submit the next. And then here is like a rough summary of the cost of those association tests that we've been running. For example, in the first panel, I'm showing the average cost of running each phenotype, so running association for the ACAF variants per phenotype. So as you can see, to our expectation, because EUR is having more sample and also more variants, it is more expensive than the other ancestral groups with about $4 per phenotypes. And then all 6 ancestral groups added up to about 600,000 CPU hours and then 22,000 US dollars. Then the other 2 types of association tests are way cheaper than the ACAF common variants because they have way fewer genetic components being tested. And then as a super quick snapshot of the results, here is a Manhattan plot of the gene level result for glucose levels. And the signal here is the loss of function variance of the gig YF1 gene, which has been previously also identified from studies on UK Biobank. And in here, in our all by all data, we also highlighting some signals like associations between loss of function variance in gig YF1 with diabetes-related phenotypes, some skin diseases, and also some prescriptions related to diabetes treatments. And then as an additional benefit of this kind of comprehensive resource, we are able to compare association test results across thousands of phenotypes at the same time. So here in this page, I'm presenting a mega Manhattan plot of those gene level results for the phi codes available in the all by all data. So we have over 2,000 phi code and phi code X being tested, and we just classify those into the disease categories shown on the x-axis. And then within each category, this like for each color, it's basically a traditional Manhattan plot you would imagine. Each dot represents a gene annotation pair. So you can see how like the signals are distributed across the genome and then across all the disease categories. And then you can probably see some of our famous genes, BRCA1, BRCA2 in the genetic disease category, and also some other famous ones like the NF1 loss of function in the neoplasma category. So those all showcase that this all-by-all resource is going to be highly valuable in further boosting genetic discoveries and the development of downstream therapeutic approaches, and then in many other aspects. Okay, so that has been all about the original data and our association testing pipeline. From here, I'll be walking you through the structure of the results. So that's making it easier for people to play around with the data themselves. And as Sam just mentioned, the data, there will be also a workspace in the Workbench for people to refer to. So in the next section, I will be doing some description of the data and then followed by a quick demo in how you want to manipulate and process the data with HALE. Yeah, because all our data will be delivered are delivered in a HALE data format, meaning the HALE table and HALE matrix table. So I'll just briefly introduce HALE a little bit. So HALE is a very powerful tool for genomic analysis at multiple different scales. It enables cloud computing and also batch computing. So here's the HALE batch components I just introduced, which we used for our association tests and for producing the all-by-all results. And then there is also a HALE query part, which can be used to simplify large-scale genomic analysis and then to better work with the super huge genomic data frames. So there are two basic data structures, the HALE table and the HALE matrix table. The HALE matrix table will be looking like this. It's basically a table with an extra dimensions. So here it has a row field and column field, and also the entries defined by column and row keys. And then here the row field and column field are both like a HALE table component. And also you can just convert HALE table back to HALE matrix table or reversely. So in some details, and this is how a matrix table will be looking like. So as I just mentioned, that there are four components, and then for the row part and the column part, they will be keyed by the index you wanted to use for this data, and the entries are the structured field indexed by the two dimensions. So for the genetic applications, we can use those as how all of us presented the exome matrix table and the ACAP matrix table, where the rows are the variants, and then the columns are the samples, and the entries are the informations of the genotypes. So here is a quick snapshot of those genotype data stored in matrix table. You'll see for each entry is defined by a sample and a genetic location. So the rows are the information for this variant specifically, and the column is basically just a sample ID, or you can also annotate some additional sample information there. And the entries are genotype, depth, and genotype quality, etc. But beyond genotype data, the matrix table can also be used to store summary statistics. So in this case, the rows are still variants or genes if we were like storing the gene level results, but the phenotype this time could be the column this time could be the phenotypes. So in this case, the entries will be storing those association test results, which are the betas, standard errors, and p-values. And then here is a snapshot of this use case, where you have the rows as the genetic components, column as the information for the phenotypes, including their number of cases, number of controls, and their types, and the entries would be everything output from the association tests. So from here, as a quick recap of our pipeline, we generated three types of association tests, which are for each ancestry groups and per association test type. We store those results in the matrix table. So we ended up with 18 ancestry specific matrix tables in total. And then later we merged those 6 matrix tables per test type into one array structured all genetic ancestry structure matrix, and then use that to implement our meta-analysis and ended up with 3 additional meta-analysis matrix tables. So this is how a matrix table of the single variant association test result will be look like. So again, the rows are some random variants, just used as demo, and then the columns are those phenotypes available in this data, and then as you can see, you can store all kinds of annotations for both the phenotypes and also the variants, and then the entries are stored as the association test results again. One thing to notice is that we also included some quality control flags in here in case the users wanted to start with the most high quality results. So here we have a collection of quality flags, which is our recommended flags to filter out variants with relatively lower quality of association tests. And then this and then the quality flags lambda is like a even stricter QC criteria on top of this quality flags. So if you wanted to apply a more strict filter on this data set, you will have you will want to use this. So let me reference this. So we have this high HQ variant flag, which is like indicated by the flags included in quality flags. And then this HQ variant lambda flag is defined by everything, all the flags included in those 2 categories, which is a more strict flag than the HQ variant. So if you wanted to apply like use a more high quality data, you can think about apply this or the second one for even stricter criteria. And then similarly for the gene level matrix table, the column field would be all the same, and the row field this time would be the genes, and then it will be keyed by combinations of genes, annotation groups, which there are like 4 available, which are loss of function, missense variant, and synonymous, and also loss of function combined with missense. And then there is also a column called maxMath, which is the upper bound of variance that will be included in the group test. So there are 3 maxMath cutoffs applied, 1e minus 2, 3, and 4. And in the entry, there's not only just one p-value in beta, there will also be 3 types of association test result available, which are the p-values for schedule test, burden test, and the SCED test. And then we also included log 10 of those p-values, and then the beta standard error for the burden test, and also related information. Similar to the previous the flags in the previous slides, we also did this for the gene matrix tables. So again, the HQ gene flag is like the recommended flag for the users to filter to a high quality set, and then it's defined by the fields included in the quality flags structure. And then there is a stricter flag called HQ gene lambda, which is defined by all the flags included in those 2 structures. And then in order to implement our meta-analysis, we first combined the results for each type of association testing into one matrix table. So as you can see here, for the GWAS matrix table, you have not only just one single column per phenotype, there will be like arrays storing all the information corresponding to the ancestry groups that have this phenotype available. So in this case, the row field will still be the same as the previous ones, but then in the column, you will have the phenotype information for each ancestry groups, and then also the summary stats are wrapped into arrays for easier computation. And then this is how the merged gene matrix table would look like. Only thing different would be it's wrapping up the burden test results, and also the row field will be the gene level information. So from here, we do some computation, wrapping up the information within arrays, and then using the array of summary stats, we compute the summary stats for the meta-analysis. And then from there, we'll have a meta-analysis matrix table where the column will still store all the information for each ancestry groups per phenotype, but within the entries, it will be a single value for each association test type, each type of association tests. And then in addition to the all those matrix table I just described, we will also provide the results in hair tables. So for each phenotype and ancestry group combination, we will provide a single hair table for each type of the results. So for the single variant level association test result, the general schema will look like this. And then this is how the gene-based results will look like in a small hair table format. So this will make it much easier for you to query just all the association, all the variant summary stats across one phenotype. Okay, so in summary, this is the number of results being delivered with all the phenotypes together times 3 association tests. We will deliver about 3,700 individual phenotype hair tables, plus all those matrix table I just described, which has 18 ancestry specific matrix tables plus 3 matrix meta-analysis matrix tables, which in total would add up to 21. And then those data will sum up to like about 70 terabytes. And then here is a quick summary of the number of stats, which is basically just the number of entries available in each of the matrix tables. So you will be able to query across, I don't know how many this is, but many summary statistics available across all the phenotypes, all the ancestry groups, and the whole genomes. Okay, next I'll be quickly showing some demo in how you can basically process a filter to the data you want from our all by all results. So first, if you wanted to load the data, there will be several things you wanted to specify. Is first you want to know whether you want to look at the hair table or the matrix table. And then which ancestry labels you would want to use, like whether this phenotype, which ancestry group this phenotype is from, and also which type of results you wanted to use. So we will have a function that can be easily used to call the path to the hair table and matrix table. So for the hair table, you will want to use the read table function, and then just input the absolute path to the file you wanted. And then for the matrix table, you will want to use the read matrix table function where you will have to specify ancestry and the test type. And then just a note that when you are using the hair table, you will want to know what phenotype exactly you wanted so that you can ask for the correct path. And then first, let's start with the how we wanted to create per phenotype health tables. So this is an example for the ACAP results. The exome variant health table should be pretty much similar to this. So on the left-hand side, you read the table and you can use describe to see what are the fields included in this table. And then you can do some filters, filter to the variants or whatever is of your research interest, and then select certain fields that are more like relevant. And then use dot show, and then you can specify the number of rows to print. And then here you can quickly examine the number of those numbers, those summary stats for those variants that you asked for. And then for the gene level matrix table, the functions are still the same, but I'm just doing some little bit more like functions here. So again, you can read the table with the same function and describe to see what are the fields included. And then you can check the number of records with dot count. And you can see there are like 200,000 records stored in this one table, which is the AMR height gene level result table. And then you can use the HAIL aggregators to count number of certain filter criterias. Here I'm counting the number of records with annotation loss of function, and then with using the maximaf of 1e minus 3. And then there are also like many other aggregators. You can take a look at hail.ease, where, for example, you can use counter to check the number of results in each functional annotation category. And then you can also use summarize to check the distribution of certain fields included in the table. And then next, I will introduce some small steps in query in the matrix table results. But just a quick note that for results on one or very few phenotype, it is always more it's always more efficient to just look at the Hill table instead of using the matrix tables. And the matrix table are mostly more helpful when you're trying to look at associations of one or a few genes across many phenotypes, or you're just trying to compare all the associations of multiple phenotypes. So here I'm taking the matrix table of the exome level variant result for EUR ancestry groups. So you can still use the same function described to see all the fields available in this in the matrix table, and then use dot columns dot show you dot columns you will be able to obtain the column Hill table of this matrix, a matrix table, and then dot rows is like grabbing the row fields for you from this matrix table. And then you can instead of just using a dot filter, you will have to specify which field you're filtering on. So there are filter rows, filter columns, and then filter entries. So you can all use to apply on the matrix table. And then you can select the fields you want and use dot show to see whatever that's of your interest. And then for the two cases I just mentioned, if you wanted to compare several phenotypes all together, you can just simply filter the matrix tables to a list of phenotypes by using this filter column function. And then if you wanted to make it convert the matrix table into a smaller Hill table, you can use dot entries, which will just explode the entire matrix table into like a long formatted Hill table. And then you can select those important fields and then use describe, you will be able to see how it looks. And then for a single, for results of a single variant components or a gene component across all phenotypes, you can first apply filter rows on the matrix table, and then select whatever is of your interest, and then do dot entry. So you can describe this in a Hill table format. And then in many cases, people might want to export the data into a text format for easier manipulation. So it's pretty straightforward. In the Hill table, you just have to do the filter and select you want, and then use this export function to like a text file path, and then you can just process this data in like a text file way. And then for the matrix table, you will have to first convert it to a Hill table first. And then here are three examples. The first one is if you wanted to just look at the information for the columns. In our case, it's the phenotype. So if you just want to check what phenotypes are there, you might want to just do this dot columns. And then if you just wanted to see the rows, you can just convert it into a row Hill table like this. And if you just wanted the entry information, you can just filter rows, filter columns, and then do dot entries to convert it into a Hill table format so that you can later apply the export function and make it a text file that's going to be easier to process in some other context. Okay. And I guess this is all about my presentation today, and I'd like to thank everyone in the team, all of us from the broad and from all of us, and especially all the participants, and then also the Hill team who enabled this analysis to become possible. And thank you all for being here.